Welcome. My name is Samarpita Roy, and I take you through a short lecture on metabolism as part of the Environmental Biotechnology course. To define life based on thermodynamic explanation entails that we explain metabolic features of cells, namely that they use water to perform metabolic reactions, carbon and nutrients to create cellular mass, generate reducing powers from electrons, and energy to do work. Another way of framing these fundamental features is to say, as Erwin Schrödinger did back in 1944, that life is organized matter which evades decay through equilibrium through absorption of energy. Specifically, when describing microbial metabolism, we can visualize Schrödinger's statement in the form of energy-yielding redox reactions called catabolism that are bioenergetically coupled in two reactions. One for the synthesis of biomass called anabolism, and second for the indispensable growth-independent processes called maintenance. The catabolic or energy-yielding reactions described in the previous slides are called redox reactions. Redox reactions are comprised of two half reactions. Electrons are transferred from an electron donor in one half reaction to an electron acceptor in a second half reaction. These redox reactions, defined as catabolism, forms the basis of microbial growth through the supply of reducing power, that is, the ability to donate electrons during electron transfer reactions. In a cell, synthesis of cellular material from simple precursors requires an input of energy. In all forms of metabolism, energy is conserved when reducing power obtained from catabolic reactions, as we described in catabolism, is conserved by the means of energy carrier molecules such as ATP. That energy generated from catabolism is coupled to anabolic or biosynthetic reactions by means of which cells grow. In this way, catabolic and anabolic reactions are fundamentally linked. To summarize, microbial metabolism in an equation where the summation of catabolic and anabolic activities define the overall growth reaction, we recognize a multiplication factor for the catabolism reaction, that is, lambda cat, which is used to signify the number of times a catabolic reaction needs to run to generate 1 mole of biomass in the anabolic reaction. Thereby, we provide a mathematical framework where catabolic and anabolic reactions are fundamentally linked as metabolism. Let us analyze the metabolism of nitrifiers using this example. We just learned to define its catabolism and anabolism. Nitrifiers oxidize ammonium to nitrate using oxygen as the terminal electron acceptor during catabolic reactions. During anabolic reactions, nitrifiers fix carbon in the form of carbon dioxide or bicarbonate and ammonium as nitrogen source to biomass. From the nitrification conversion, you can see that ammonium, the electron donor, is oxidized to nitrate, the oxidized form of the electron donor. This conversion forms the electron donor reaction of the redox couple. Next, in the electron acceptor reaction, we recognize oxygen as the electron acceptor. That, by the end of the reaction, is converted to water. This overall catabolic reaction generates energy for biomass synthesis in the anabolic reaction. The biomass here is represented as a simplified chemical formula, C5H7O2N. In the anabolism, carbon dioxide serves as the carbon source and ammonia as the nitrogen source to form biomass. A diverse range of microbial metabolism thrive in using different types of electron donors, such as organic compounds or inorganic compounds, and various electron acceptors, such as sulfate, nitrate, oxygen, or even other organic compounds, such as in fermentation reactions. So far, we have described mostly microbial metabolism driven by chemical energy. However, the catabolic reaction can also be replaced by light as a source of catabolic energy, and then this is coupled to anabolic reactions. Such a phototrophic metabolism is observed in cyanobacteria, microalgae, green sulfur bacteria, and others. The Delft School of Microbiology that you have been introduced to from the previous lecture was for a large part evaluating different microbial metabolism by combining various electron donors and electron acceptors. The one way to study these different microbial metabolisms was to establish environmental conditions that specifically select for these metabolisms. With the next example, let me show you how we can apply the ecological selection principles to study bacterial metabolism and inform biotechnological processes. Based on the metabolism, I will elucidate an example of using ecological selection to enrich for nitrifiers and denitrifiers. Nitrifiers, as we saw in the earlier example in this lecture, in part convert nitrite to nitrate and utilize inorganic carbon to fix into biomass. Denitrifiers, on the other hand, also utilize nitrite, but with organic carbon to fix into biomass. Thus, both require nitrite as substrate, but with different ecological niches. Now, with this knowledge of the metabolism of nitrifiers and denitrifiers, let us evaluate bioreactor operations to enrich for these organisms. If we operate a typical bioreactor with nitrite as electron acceptor and bicarbonate as the carbon substrate in oxygenated conditions, we will observe nitrate as a gaseous product evolve from nitrite oxidation. This conversion is part of the nitrification process. Therefore, such an enrichment culture will enrich predominantly for microorganisms known as nitrifiers. Alternatively, if we make small changes in this bioreactor operation, that is, provide nitrite as electron acceptor with an organic carbon substrate under anaerobic conditions, then we observe nitrogen gas evolve due to a process known as denitrification. In such an enrichment culture, we will preliminarily enrich for denitrifiers. The process of converting ammonia to nitrite and then nitrate, as we just discussed, is known as nitrification, as shown in the blue-colored arrows in the figure. It is frequently applied in wastewater treatment to convert large amounts of nitrogenous waste to nitrate. This nitrate, in turn, serves as the electron acceptor for denitrification process, where the final product obtained is nitrogen gas, as shown in the orange-colored arrow in the figure. This ensures that the nitrogenous waste that enters a typical wastewater treatment plant is converted to a benign form, that is, nitrogen gas, and released back into the environment. However, it is important to remember that the design of this process and its subsequent improvement have been a result of understanding and applying concepts of microbial metabolisms.